Thanks, Matteo, for a very nice introduction to my talk, even though it was perhaps not planned. So my talk today will be quite empirical, no computer vision, no fancy machine learning, old-fashioned methods, or before I was aware of all these wonderful tools we now have. And this is some work I've been doing on dragonflies and ostriches, some which is published, some which hopefully soon will be published, and I have a lot of other data, more comparative data that I will not talk about here. But it's about thermal imaging as a tool to study quantitative genetics and selection on plasticity and thermal adaptation. So first, a bit general reflection here. A lot of the talks in this conference have been focused on morphology, and that's quite natural, understandable, because we can easily or more easily quantify morphology from museum collections. There are specimens. And then we make inferences about ecology and evolution based on these morphological measurements. But it's important, and I don't think anybody would disagree, that morphology is not enough for phenomics. Phenomics also encompasses physiology, behavior, and other traits that are not necessarily expressed in the external morphology. And this is also one reason why we should never use the term living fossil that you often hear in grant applications and sometimes journal articles. Typically when we talk about or when others talk about living fossils, we typically point to the coelacanth, this deep sea fish, or the nautilus, or even dragonflies. I'm guilty, sometimes have used it also, that we have these wonderful fossils of dragonflies 150 million years ago, family Gonfida, and those which live today are very similar. So we say, we might easily say, they haven't changed. But as a matter of fact, these and other so-called living fossils have often changed if you go beyond the morphology. And another aspect why it's important to keep in mind is that many traits, particularly physiological traits, but also morphological traits, are plastic and change with environment. Here is a picture of a thermal image of a damselfly we worked on at one temperature, and then the same individual as the temperature heats up and it gets warmer. So plasticity is also important, whether for morphology and other traits. And another reason also to be cautious and avoid the word living fossil is that if we look at the genetic level, genomic level, for instance, this study on the nautilus here, on a series of populations that looked very similar, if you look at external morphology, there were deep genetic divergences. So a lot of presumably cryptic species. And there are all reasons to believe that's the same with the coelacanth and these other organisms. So, but we still have a morphological bias, I would say, in evolutionary biology. And here is one illustration of this. This is an important paper, a wonderful meta-analysis by Kingsolver et al. More than 20 years ago, where they did compile all the selection estimates in natural populations. And they found that most selection estimates were from more than 80% of all selection estimates from natural populations were morphological traits. And there were very few selection estimates on behavioral and physiological traits. So I think it's more important than ever that we try to think about these other non-morphological traits, and particularly traits which are plastic and change during the course of ontogeny. We can also flip the whole question and think about there are many cases where populations and species are very variable in their external morphology. This species sitting here in front of me is a good example. I see a lot of diversity in skin colors, maybe not the entire diversity of the globe, but a fair amount of diversity. And we know some large part of this variation is explained by latitude, generally darker skins in humans near the equator. But interestingly, if we go inside our bodies and measure our body temperatures, it's almost no variation at all. Almost all humans have 37 degrees Celsius inside their body. So there's an interesting disconnect between the external diversity but internal similarity. And this puzzled the late evolutionary biologist George Williams a lot in maybe his best books from '92 about natural selection. He thought this was one of the major mysteries and most fascinating problems in evolutionary biology. And it's not only true for humans, it's also true for mammals and birds in general. He thought it makes no sense. Why would mammals have the same body temperature whether they are Arctic poles or in the rainforest or in the desert? Why wouldn't selection favor some local adaptation to local conditions? And he speculated maybe there is no genetic variation in body temperatures. So maybe it can't evolve. So there would be some kind of hard constraint, no lack of variation. And he suggested, to my knowledge, this has not been done, but he suggested you should do an artificial selection experiments on, say, mice, short-lived, where if you found different families with different body temperatures, could you select and create different mouse lines with different body temperatures? To my knowledge, that has not yet been done. But mostly, I feel, have very big problems in accepting there would be no genetic variation. In the literature on ectotherms, animals like insects and reptiles, where the body temperature is dependent on the external environment, there has also been a lot of focus on how body temperature changes. And some famous work by herpetologist Bogert in the 1940s, followed up by Raymond Dewey and Marta Mundos and others, have looked how thermoregulation can buffer thermoregulation and behavioral plasticity can buffer organisms from temperature-mediated selection. Namely, Anolis lizards and other lizards, they move around in their local environment. They go from hot spots when it's hot outside to cold spots to cool down the temperature. When it's cold outside, they seek out hot spots. Thereby, they buffer selection from changing their physiology, meaning that behavior plays a conservative role in evolution, slowing down physiological evolution. So this is an interesting case where behavior acts as a conservative force and slowing down phenotypic evolution. So to study all these are fascinating problems. They're also very difficult because body temperatures is difficult to measure. Thermal imaging, infrared cameras, is a non-invasive tool where you can quantify individual variation in thermal physiology, assuming that there is a correlation between the external body temperature, the surface temperature, and the internal body temperature, which there typically is. The strength of this correlation varies, but there typically is. So here are some nice examples of animals from Glenn Tattersall's lab, measured in more or less similar environments. And I've been interested in this from the perspective of the evolution of environmental tolerance and plasticity, building largely upon the framework by Louis-Miguel Chérin, Russ Lande, and the late Georgina Mays, where they have developed a framework where we think about reaction norms, that is how a trait plastically changes in relation to the environment, and then connect that to the adaptive landscape, how fitness affects the changing trait values, and then thinking in terms of tolerance curves, how fitness in its turn is affected, changed by the reaction norm slopes and intercepts. And here, one of my study organisms are these damselflies, two species we have in Sweden, Calopteryx splendens and Calopteryx virgo. Here are the males. They differ strikingly in the amount of wing pigmentation. Calopteryx virgo has entirely dark wings. Males in splendens have only 50% of the wings. And we have shown, using various experiments, that the wing color is target of sexual selection. It's also important in species recognition by females. And interestingly, the more melanized species has a more northern distribution, and it's also darker. That fits quite well with functional arguments that melanin not only functions in sexual selection, but also has a natural selection consequences. And looking at survivorship and mating success of both females and males, broadly speaking, mating success, the optimum for mating success seems to be at higher temperatures. At higher temperatures, mating rates increase, but at higher temperatures, survivorship decrease. These are mark recite studies in natural populations. So broadly speaking, if they want to survive long, temperatures outside should be low, but if they want to have a high mating rate, temperature should be high. So there is a conflict between survivorship and mating success. We have measured individual variation in body temperatures using thermal imaging, taking a repeated photo with a few seconds in between of hundreds of individuals of both species. And here you can see how they walk. First we cool them down under a standardized condition, then we take multiple pictures of the same individual and see how fast they heat up. And then we quantify reaction norm slopes, how fast they heat up, and intercepts. And here are the species, and here are the mean reaction norm of the two species and the slopes. And to make a long story short, the slopes, the plasticity, does not differ. They increase in temperature about the same rate, but the intercept is higher for Calopteryx virgo. Here the scales are unfortunately displaced, so it looks the other way around. But this species has a higher intercept, higher body temperature at low temperatures, but individuals increase at the same rate. And then we have used these data on individual variation in reaction norm slopes and intercept and estimated sexual and natural selection gradients on these traits. And again, generally speaking, it seems like sexual selection favors steeper slopes and higher intercept, and natural selection is the opposite, lower intercepts and lower slopes. At least for this species, it's somewhat different story for Calopteryx virgo. But these intercepts and slopes appears to have fitness consequences, and here we have visualized it in relation to external temperature measured at the stream. So here you can see, for instance, longevity, survivorship, in relation to the slope, and it goes down with steeper slopes and higher temperature. Longevity gets smaller, so they survive less well when it's high temperature outside, and when with those individuals who have a steep slope. So selection seems to favor more shallow slope, especially at high temperature. And then in mating success, at least for virgo, it appears to be the other way around. Mating success increases with the intercept and with temperature. Okay, from insects in Sweden to southern Africa. These are ostriches from South Africa, which I've studied with some colleagues in Lund and South Africa. And these are very large birds in Oudtshoorn in the Western Cape province. There is a semi-natural populations, a pedigreed populations we keep track, or not we, because there is a whole crowd of specialists to capture and mark these birds. I'm glad. I've been doing field work, but I have never touched them. I don't want to, because they can easily kill human beings. Anyway, I walked around, my most bizarre field work I've ever done, walking around with an infrared camera, taking photos of these. The problem taking photos of infrared photos of these is that they are run towards you, and then they fill up the whole camera. And there are also barbed wire, and you feel very small when they run to that, and there is literally only a barbed wire between you and an animal that can easily kill you. Anyway, we did this and caught a lot of thermal profiles of different individuals over a number of years. This study started actually almost 10 years ago. And we looked at how different body parts change in temperature in relation to the environmental temperature. And we found that the neck appears to be more sensitive, it's more plastic to the environmental ambient temperature than the head. So the head is kept more cool. And the difference between head and neck temperature is that as the neck increases in temperature, the head doesn't increase as fast. So they keep their head cool. And this also has consequences for reproductive success. At hot temperature, this head-neck difference, if the head is cool relative to the neck, reproductive success increases. But you see no effect at benign temperatures. And finally, we have also estimated repeatabilities and heritabilities and evolvabilities and additive genetic variances of thermal profiles. And it turns out there is significant heritability and repeatability, so there is potential for evolution to change these thermal profiles. And finally, we have also compared in this area, a common garden environment in South Africa, we had different subspecies, and it turns out they differ in the degree of plasticity. The southern subspecies appears to be able to cool its head more during heat waves than the tropical Kenyan red. So the southern subspecies come from a more seasonal environment and seems to have evolved the ability to cool the head more in relation to heat waves, unlike the tropical subspecies, which has a red neck in Kenya that experience more stable temperatures. So just to summarize, I think we need more phenomics research and selection studies on quantitative-genetic studies on physiological traits. Thermal imaging could be one such tools to collect these large amounts of data. And I think that these data from insects and ostriches show that thermal traits can be targets of both natural and sexual selection, and they are also heritable. And I think one major challenge in phenomics is we need to increase the speed and efficiency by which we analyze these thermal images. Here we have paid a lot of students over the years, but that was quite expensive. And with this, I'd like to thank my collaborators and the funders, especially in the ostrich study, Mads Schuh, former postdoc in Charlie Cornwallis' lab. The only time I can have Carlsberg Foundation on a grant of funder. It's not the best beer in the world, but Mads was a really good postdoc to collaborate with. Okay, thanks.